So hi everyone, I'm Remy. I am by training, I'm a biologist, and I use what I tell everyone is that I wanted to understand how the brain works, and now I spend my time telling people how to name their files and organize them. And that's why I'm here today, to tell you about how to name your files and organize them, because that's what the brain imaging data structure is about. I'm also I'm actually one of the BIDS maintainers. There's a group of people who are in charge of maintaining BIDS. I'm one of them, and I'm also a maintainer of NILEARN, and I'm part of the Oregon lab at DMNI. So BIDS, this is what's going to be on the menu for today. So first, sort of why BIDS at all? Then I'm going to tell you a little bit about the data management principles that underlie BIDS, and then I'm going to go and try to give you some tips and pointers on how to make your life easier when you're working with BIDS. Okie dokie. All right. So why standardized data at all? So this is a table that was taken, I think, from a school book from, I don't know, I think it was in Vienna from mid-19th century. And then we're just comparing units across different countries in Europe. And you can see that in red, you have different countries that use the foot as a unit. And you can see that even though each country uses what they call 1 foot, the actual length of it varies quite a lot compared to the Austrian foot, because that's a table that was produced in Austria, right? On the other hand, what you can see is that all the countries that actually used a meter tend to have the same value for each of the measurements. And I always try to imagine when people were doing science back then, they would sort of have to first send each other their data and then sort of agree on which foot did you actually use for all of this, right? So sort of standardizing and sort of starting using the metric system kind of helped sort of reduce friction when people wanted to communicate and speak sort of the same language when it came to science, but also other things, obviously. You may think, okay, well, that was like a century and a half ago, or maybe even 2 centuries ago. That's really not an issue anymore. Well, that's from some of you maybe know that in 1999, a lander crashed in Mars because 2 of the teams didn't agree on which unit they were working with. One part of the team was working with a metric system, and the other one was working in feet. And then obviously there was some calculation error in the end that resulted in just something crashing on Mars. But we're not trying to land rovers on planets. We're only trying to understand how the brain works. So obviously that doesn't matter to us, right? Well, maybe actually it does. So I'm going to start a bit with a question I tend to ask people, is that should you stop doing sort of good research data management if you're not using a BIDS data set? Let's say that you have another data set that has nothing to do with brain imaging, and should you stop applying some of the principles that people who do good research management tell you to apply just because it's not a BIDS data set? You may say, well, obviously not, but actually every time I tend to give a BIDS data set to someone and they start analyzing this, after usually a few hours of just working on it, then you start already having data everywhere, and the file naming tends to sort of drift. And then after 6 months, it's a bit of a mess. So the idea is that you may start from a situation when you start a PhD or when you start a new project, when you have like a blank slate and everything is perfect and pristine, and you know that this time you'll get it right and you won't end up in a horrible situation in terms of code and data in 6 months. But what most of us, when we do research, we want is we actually want to answer a scientific question, right? We don't want to be spending time thinking how to organize data and actually organize the data and annotate the data, right? So we actually just want to get straight to our answer. So we tend to rush, and then after a little while, the clean slate that we had at the beginning ends up being kind of a bit of a mess, and then we sort of are lost within our data, and we just don't want to even work on it anymore because we don't know how to find the information we're actually looking for. And that's pretty much the PhD situation. When you're a postdoc, that's pretty much you have to juggle with 5 projects at the same time. So if you've not got good habits on how to organize your data in general, then you end up just in a pretty sort of, let's say, interesting situation. So yeah, pretty much BIDS and in general data management when it comes to, and many open science practices are there to help yourself, right? So cleaning your data will make it easier to share them with your closest collaborator, which in this case is yourself in 6 months. And so, you know, why standardized data and why curate data? That's one reason, but then why BIDS? Well, BIDS sort of came there were plenty of reasons for it, but one of them was try to get away from what we call the N+1 standard problem, which is sort of illustrated by this comic, XKCD comic, where, you know, you start with a situation where you have 14 competing standards for, you know, something. In this case, it could be, I don't know, just instant messaging or anything that has to do with like encoding of characters. And then people get together as like 14 standards. That's ridiculous. We need to have one of them that only one that covers all use cases. And then soon after, you end up with 15 standards, right? So we wanted to sort of avoid the situation, and BIDS tries to sort of tackle most use cases, but not all, and try to make sure that it's a standard that's easy to adopt for people so that they're not going to go and try to create another one. Okay, so I've told you that it's good to organize your data, even if it's not, you know, neuroimaging data, but then I should tell you a little bit more about what the data management principles that underlie BIDS are and how you can sort of start applying them even when you're not using a BIDS data set. So let's just step back a little bit. BIDS stands for Brain Imaging Data Structure, in case you didn't know, and it's mostly about FAIR data. So FAIR stands for findable, accessible, interoperable, and reusable. And BIDS is mostly about the last two, so interoperability and reusability. So interoperable tends to mean that you're going to be using data formats that are easily going to be easily read by most softwares and going to be going to work well together. And reusability means that if I give you a data set, it is sort of standalone and self-contained enough that you're not going to have to sort of send me a million emails to understand how it's organized. So all of this sort of relates to, obviously, yeah, data standardization and research data management, as I was saying. But more specifically, BIDS tries to do a few things. It tries to sort of forces you to modularize your data. It forces you to specify a folder structure, and it gives you sort of strict rules on how to name your files in a way that's friendly for humans and also computers. It relies on using standard sort of interoperable file formats. And BIDS gives you plenty of ways to sort of both document your data and also tries to minimize duplication of information about your data. Okay, I'm going to sort of go through all these different steps and give you ideas of give you more details about each of these different points now. Oh, and very often you'll see those gray messages. If it's gray, that means like that's pretty much kind of a take-home message. And if it's red, it pretty much just means just please don't do this. Even if you're not working on a BIDS data set, that's usually kind of a bad practice. Okay, so first I'm going to tell you about how BIDS sort of forces you to modularize your data. And I tend to sort of explain this with pasta. The reasons that in the previous lab where I was, there was about like more than 50% Italians, and the only way I ever found to sort of, you know, get the message across was to go and explain it with pasta, because it got to sort of their national pride, and then eventually the message sort of sunk in. So raw data is, let's say, almost the rawest ingredient. No, source data, sorry, is like the ingredients that you start with to sort of, you know, when you want to make a recipe. So in this case, would be like the tomatoes or like the vegetables that you're going to need for the sauce, the flour, the olive oil, and so on. Some of you may notice, well, actually, flour is already kind of processed. That's kind of true as well for a lot of the data we actually acquire. For example, in an MRI scanner, most of us will get DICOM data. We're not going to get the K-space data. So even when what we call source data, so a lot of this might already be slightly sort of processed. Then we have what we call raw data, where the source data has been sort of massaged enough so that it's easier to reuse. And you can do many, many things, but you're not constrained so much that you can only do one thing, right? You can do many, many different recipes, even with that kind of raw data. And then you have derivative data, which could be either sort of like pre-processed data or even statistical, like the end result of statistical analysis. And this would be sort of final recipe that you have at the end of your cooking, right? And this is what, when someone sort of publishes a paper and they give you pretty pictures of their final results, this is, when you ask for data, this is sort of what you expect from reading the methods section, right? And then what the data set actually looks like, if you ask for the data and they send them to you, very often it's actually a big mix of everything together, where you have source, raw, and sort of derivative data all together, all at once. Sometimes you may get lucky. There may be a README, but a README on this kind of data is like hoping that adding Parmesan cheese on all of this is actually going to make it easier to sort of digest. So in case you're wondering, I actually had to eat some of that after I took the picture. Do not recommend. So please organize your data and modularize your data. So in the case of BIDS, what it means to modularize your data is that you're going to have, you have your source, your raw, and your derivative data. BIDS is mostly concerned with raw data, and it says a few things about derivatives, but very little at the moment. And it says pretty much nothing about source data. So you can organize your source data however you want, but when it comes to your raw and your derivative, BIDS has a few things to say. And this would be sort of the please don't do this example. So if some of you have worked with a software called SPM to analyze your data, by default, SPM will always dump the derivative data wherever it found the raw data that you gave it. So it becomes really hard when you work with softwares like this that behave like this to sort of make sure that your data is properly modularized. So yeah. Take-home message: don't put your derivative data in the same folder as your raw data. Then BIDS will sort of tell you how to organize the folders in your data. So how many folders, too many, like do you want many folders or you want very few folders? You have to find the right balance. On the one hand, too many folders make it very hard to navigate the data unless you really are a fan of clicking a lot. And a single folder where you dump everything becomes really hard to sort of find things without having some sort of crazy file naming scheme. BIDS tries to strike a balance between the two, where you're going to have a subject folder for the subject, an optional folder for what we call session, and then another folder for each data type that you have in your data set. And then the data type, the actual folder naming goes that every subject folder starts with sub, every session folder starts with ses, and then each of the data type folder sort of gives you a hint of what type of data you're going to find in each. So ANAT will be for anatomical and structural MRI, for example. Behavioral data will go in the BEH folder. Motion data will go in the motion folder, and so on, right? And those are sort of grouped under sort of what we call modality. So either that usually tends to point to what kind of sort of device was used to record the data. So MRI, PET, MEG or EEG, behavioral data, microscopy, near-infrared spectroscopy, and motion. So these are all the different modalities that are actually covered by BIDS officially at the moment. But if your favorite data type is not in there, be aware that there are extensions of BIDS in progress, and that you can sort of already start organizing your data by following those extensions if you want. Okay, so nope, yes. Okay, so after you sort of organize your folders to know where you're going to put your data, you sort of have to sort of figure out how you're going to name the different files you put in it. And BIDS tries to also follow some of the general rules about good sort of data management. One of them, the first one would be just don't put any white space in your file names, because it makes scripting very, very hard. Try to stick to only using letters, numbers, hyphens, and underscores for your file names, because some operating systems don't handle them well. And also, if you're going to have to write those wonderful things called regular expressions and you have special characters in the middle, then you're in for a lot of fun times, usually not so fun, actually. Then next rule is don't rely on the letter case, so uppercase or lowercase, to sort of distinguish between two files, because some operating systems don't make a difference between, for example, an uppercase A or a lowercase a. I think if I remember Mac OS or some older version of Mac OS, I think they've actually changed more recently, or don't make a distinction between those two things. And then use separators in a meaningful way. So use underscores and hyphens in a consistent manner. So you have what are called cases for either file names or variables in your code. And some of the main ones you may find around are like camel case or snake case. And sometimes you may even see something called kebab case. There are some others, but those are the ones you would see in a BIDS dataset, for example. Okay, but then comes the question, thanks, you've given us all these sort of very generic advices about, how to sort of good practice when it comes to naming files, but, what's actually is the actual rules that you use in BIDS to create a file name? Well, so that's pretty much, it follows, they all pretty much follow the following pattern. So you start with, from all files with an extension, a dot and the extension. If you're, for some reason, your file browser on your computer don't show them, just change the options and you should always see them. And then right before the extension, you've got what we call a suffix that is preceded by an underscore. And then before that, we've got a series of what we call entities, and each entity is composed of a key and a value, and then they are separated by a hyphen, and then each entity is separated by an underscore. So, and yeah, the next rule is that all of those things, key, labels, suffixes, can only contain letters and or numbers. But that's pretty much the general recipe to construct a BIDS file name. So even if you, and what I tell, and what we say in BIDS is that even if you have to add new files in your data sets or generate derivative data from a BIDS data set, try to always follow this file naming pattern, because then you can always reuse some of the sort of tooling that exists in BIDS to work with this file name sort of pattern in general. Okay, and so that was sort of the abstract example, and this is what it concretely looks like. So here, for example, you have a very simple sort of BIDS data set with one subject, with your sub01 folder, your ses01 folder for the session, and then you have two data type folder, one ANAT for structural MRI, and the other one EEG for EEG data. And you can see that the suffix at the end sort of is usually a good proxy for what type of data is in this file. So in this case, T1W stands for T1 weighted. You have the EEG suffix where the EEG data actually is. The channels is where you're going to find all the information about from which channel the EEG data was recorded, and events sort of tells you what happened when during your experiment, right? And you can see that there are different types of entities you're going to find in the file names. So the sub entity, the subject entity, and the session entity are repeated from the folders. And then, for example, as soon as the participant was doing something, you're going to have a task entity. And then if you have several run of this task, you can also, for example, use the run entity. And if you go through this BIDS specification, you're going to see a very long list for each sort of data type. What are the sort of entities that are obligatory, or some that are, or which ones are optional? So for example, for EEG, subject and task are obligatory. So, and any of the others that are between square brackets are optional. So if you wonder how, I should name this very specific files, you should sort of refer to the BIDS specification and look for these sort of file name templates to know how to name your files. Okay, so that was the file names, and now about file formats. I was saying that in general, the rule is try to use sort of standard format that are interoperable. In the case of BIDS, that these are the sort of the formats that we tend to use for the different data types or modalities. So for MRI, we use Nifty files. For surface data, we use GIFTY. PET also uses Nifty. EEG, MEG, and intracranial EEG have a whole plethora of formats they support. I'm not going to go through all this because it's actually it would just get actually pretty long. If you have microscopy data, it's either PNG or TIFF or their OME equivalent. And near-infrared spectroscopy relies on the SNIRF format. So that's for like the data related to each of the sort of imaging modality that you're working with. But if you're working, when we're talking about things that are sort of text data or tabular data, the general rule is stick to data formats or to formats that can be open in a very simple text editor, right? Where you don't need a license or the software is not proprietary in any way. So usually that means stay away from doc file for Word, for Excel files, for don't save your data in a mat file if you're working with MATLAB and similar cases. And that's why when it comes to how you save tabular data in BIDS, we use what's called a TSV file. So tabulation, separated value, and that's for spreadsheets in general. And the rule in BIDS is that you must include a header, meaning there should be a line at the top telling you what each column is about, or at least having a name for each column. And we say that you should use snake case for the headers. So for example, here we have an example, a bad example. This is when you work with fMRI data, you may need to do some motion correction and realignment on your data. And this is the output of this step that is given by SPM, which is one of the main software for statistical analysis of fMRI data. The good thing about this is that it gives you a text file, a .txt file, so you can open it with pretty much anything. Any file editor will should be able to open this. The problem is that amongst these columns, you have 3 columns that represent rotations and 3 others that represent translations, but you have no idea which one is which. So you get good points for having a file format that's easy to open, but the user has no idea how to reuse this data because it doesn't have enough information about it. So that's why we tend to say, we say tabular data and BIDS must have a header. And this is what a participant.tsv would look like, for example. So participant.tsv is a TSV file that's at the root of your data set, where you can sort of put all the information about the different participants in your data sets, whether it comes to sort of age, sex, because BIDS supports different working with animal data. You can have species, which group the data was each participant belong to, and many other things, right? And this is pretty much what this would look like if you open it in a file editor. And obviously you can still use Excel to sort of like load this, and this would just look like a regular table. Okay, the other type of files you're going to find in a BIDS data set are what's called JSON files. So unless you worked with BIDS before or you've done a bit of programming before, you may never have heard of JSON files, and it stands for JavaScript Object Notation. And because JavaScript is a coding language used a lot on the web, and the standard in JavaScript is that most of your variables are going to be in camel case. So that's why if you open any JSON file in a BIDS data set, you're going to see that you're going to see a lot of things written with camel case. And we use those in BIDS to store information that go in pairs. So where you have an attribute and a value associated to it. And the way they also function in BIDS is that they're called sidecar files, meaning that you have a data file, let's say an image, a nifty image of a brain, or an EEG data file, and next to it, you're going to have a JSON file that contains the metadata about this data file. So here that would be in this case for some bold imaging data, or for the events file that goes with it, you have a JSON file next to it that contains extra information and for the data file. And when you open it, this is what it would look like. This would tell you, well, for this sort of nifty file, the task name that it relates to is an NBAC, and the repetition time for this file is 0.8 seconds. And this is where you're going to sort of store all extra metadata for your files. So this is sort of one of the bad example. This one actually comes from a whole bunch of data sets that we have in the BIDS example repository on GitHub. Sometimes I wonder whether we call it BIDS example as in good example or bad example. I still haven't figured out why. I would say that this one is a bad example. Because someone wanted to put a lot of information in the file name about what the task was about, so they called it task living non-living decision with plain or mirror reverse text, right? And this information is partly duplicated in the JSON file that goes with it. A better way to sort of use those JSON files to record metadata about your experiments in general is to do the following: stick to a much shorter name, for example, here for your task, just call it decision, and then use all the extra fields that BIDS provide you to add more information to your data set. So here you have a task description field that allows you to put as much information describing the task and what it was about. You can also, very often in BIDS, you can use pointers to what are called ontologies that will give you a very strict definition of what type of task, for example, it was that people were doing. And here as well, you can sort of, if you gave your participant a very precise instruction, you can also copy them in the JSON file of your data set. So this is what I would tend to tell people: just make sure that you add all the metadata you can in your data sets, because it will prove useful in the long run. Okay, so we're getting close to the end of sort of the different parts of what are the sort of research data management principles in BIDS. The last one I want to talk to you about is how there are some ways in BIDS where you can minimize duplication of information in your data set. So for example, imagine that you have 40 subjects and each of them had 2 sessions and 4 runs per subject per session. If you do the math very quickly, that's 320 JSON files to edit. Which, for example, you want to follow the advice I gave you right before, where you want to sort of add extra information for what the task was. So now you have to edit all those files, and you're not really looking forward to spend the next 2 days just opening each file and copy pasting things. So this is the minimal example of this, with just two subjects. And if you are using exactly the same protocol for all your participants, it's very likely that the metadata that you have in those JSON files is going to be exactly the same, right? So you end up with just duplication of data across subjects. So one way to sort of deal with this in BIDS is to actually put a single copy of the file in the root of the data set and remove sort of the sub-entity from it. And just keep all the metadata, a single copy of all the metadata for these files in the root of the data set. That's what we call in BIDS sort of the inheritance principle. The idea is that all the metadata that's related to this NBAC task that you find at the root of the data set will be inherited by all the files that you find in subfolders. So you know that and the NBAC information in here will apply to both this one and this one. And that's sort of the principle that some of you who may have heard when it comes to coding, where you should just do not repeat yourself and only say things once if you can. Okay, so I'm sort of done with the first part. Now I'm going to give you a long series of tips and pointers. It's going to be a lot of also showing you where to find information on the web when it comes to BIDS, and if you need to do very specific things. Obviously, I'll be sharing the slides, so it's not like you need to keep track of all of those. Okay, so I'm gonna jump right in. If you are completely new to BIDS and you maybe if this talk is like maybe the first thing you hear about, one of the first place where you may want to go is actually what's called the BIDS starter kit, which is sort of really tries to give you the basics about BIDS so that you are not completely lost. Aside to that, if you're not the kind of person who likes reading, but you prefer like the sort of the video format, we try to curate a whole bunch of YouTube videos that are about BIDS, either that describes what BIDS is or what you can do with it or how you can use it. Yeah, and if you hear about other videos that we have not listed, let us know so we can sort of make sure that we are up to date on this. Next place where you may want to go to get information would be the actual BIDS specification. Then sort of like, the sacred text where, that says what BIDS, the truth when it comes to BIDS. My main recommendation when it comes to using the specification is that don't try to read it from like, the very first page to the last one, because that would be almost as entertaining as reading a phone book. But try to actually just use the search bar and just, search for whatever information you're looking in there. And it should be able to sort of point you fairly quickly to what you're looking for. Or maybe use the glossary. There's a glossary in the appendix that will sort of give you sort of definitions for what you may be looking for. Then if you're looking for any resources about BIDS, what we're trying to do at the moment is to centralize everything into a single place. So we have this sort of awesome BIDS, which is just a list of everything about BIDS, whether it's about documentation, list of converters, tools about BIDS. We're trying to centralize everything in one single place. So that is usually where I would send people when they are looking for just any of those. I was talking about converters, and that's usually sort of the first thing people have to do when they work with BIDS is convert their source data into a raw BIDS data set. The problem you may have is trying to figure out how to choose one, because there are several of them. Well, obviously, choose a converter that will work with the kind of data you want to convert, whether it's DICOM data for MRI, whether it's EEG, whether it's eye tracking data. Not all converters can do all. Most of them actually tends to specialize in a single modality. But so just concentrate on those first. Then most converters tends to be written in a sort of a specific language, or will have you sort of write a few extra files in a specific language or some file format. So depending on whether you're more familiar with Python, MATLAB, or R, you may want to choose different converters. If you're more, you may have to work with a JSON file or what are called YAML files. So that might also help you to decide which converter to choose. Finally, I think one of the main things that should sort of guide you sort of to choose a converter is very simply what people around you use. Because if everyone in your lab uses the same converter, it's going to be much easier for you to find help around you if you start using this one. So yeah, that would be sort of my last advice about this. Is also try to sort of have a look very quickly at the sort of the user friendliness of the converter, how much documentation there is. Some of them have graphic interface, so maybe you want to sort of use this. So really try to have a quick look of to see which one might be the most friendly depending on your sort of technical skills. To give you sort of a pre-list, because there are many of them, of where to start looking. When it comes to MRI, I would say that the three main ones are going to be HUDICONF, BIDSCoin, and DICOM to BIDS. So they are the three main ones that you'll see around. And for EEG, there are several others. Usually, to my knowledge, conversion for EEG or MEG or intracranial EEG is usually easier than with MRI data. But I know that EEG Lab, for example, and M&E Python have some tools to help you convert your data to BIDS. Okay, So that was for conversion. But once you've converted the data, usually what you want to do is make sure that your data set is valid. For that, then you are going to be using what's called the BIDS validator. Most of you, if you've used it, you will mostly know the web site, so the web format of the validator, where you open this website, you click on choose file, and then you point the validator to the root folder of your data set, and then it will sort of make a list of everything that's in there and tell you, report some errors or warnings depending on whether your BIDS dataset is compliant or not. So you get errors if something does not comply with BIDS, or if you are missing some required information, and you get warning if you are missing some recommended information, for example. You may just use the website for the BIDS validator, but in case you're working from the command line or on the computing cluster, there is also a CLI tool that you can use from the command line, also a containerized version of this that you can use with either Docker or Singularity. And one of my converting tips when it comes to working with BIDS is that you convert one participant or very few participants, and then you validate your data. Do not try to convert a whole bunch of participants or a large amount of data and then only validate, because if you have, because you may end up just being drowning in errors and warnings, and it will take you a long time to troubleshoot where they came from. So do your conversion really step by step and bit by bit, so it will be easier to handle. My next advice is obviously fix all the errors, so all the file naming, all the missing metadata, but also try to make sure you have as few warnings as possible, because warnings are there for a good reason. And they usually point that you're missing some data that, even if it's not required, will be actually very valuable for others or even yourself in 6 months when you want to sort of have a look at your data and figure out some specific details about it. Next thing I would add to this thing is that before you finish validating, once you're done validating and you go back to converting more data, I would say try to use a version controlling system to sort of make a checkpoint of your where your data was at this time. I think later during the term, I think it's either next month or in December, I don't remember what you'll hear about data. Next month?
December.
Oh, in December? Okay, yeah. So you hear more about data lab in December, but I tend to recommend that once you have a valid data set, before you go on to converting more data, make a save of it using data lab, because then you have you can always go back to in the history of the life of your data set, you can always go back to this point where your data was valid. Okay, so now I want to tell you a little bit about just annotating your data and give you some pointers to some tools that exist. So annotating data is important for several reasons. For example, yeah, I could tell you that if you have a minimally valid BIDS data set, with the current level of automation that exists for fMRI data, you can pretty much get from your raw data set to your statistical maps with, more or less 2 commands and 1 config file, and you can just do everything, right? But if you don't have good metadata, you won't be able to interpret, to make any interpretation of those information. I mean, if you are the person who acquired the data, yes, you may, but if you give this data set to someone else, you might say, oh, I have activation from this condition compared to this condition in this part of the brain. But if you have no idea what the condition actually is and what the people were doing in the scanner when they were seeing something on the screen or listening to something and what was exactly happening, you won't be able to actually make any interpretation about your results. So the rule of thumb for this is usually try to just enrich the data set, annotate, add more information to the data set with more contextual information just to help the final interpretation of the results. And there are very simple way to do this. The main and most obvious one is just add a lot of information in the README of your data set. There is a tiny README in the BIDS starter kit that you can use to sort of know what extra information you can put in there to help others reuse your data. Or even just yourself, because there are so many details and so many things that happen during data acquisition you may not remember in 6 months, that just added to the README that will be useful. The other part of your data set that you can annotate, and it's really important to annotate, would be the participant.tsv that I was mentioning earlier. For example, here you have 2 columns. You have columns that relate to age, sex, group, and handedness. But if you are just given this with no participant.json file where you have description about what those columns means, you have no idea whether age here is it in, are we working with adults and it's years, or are we working with animals and it's and there are months? Like, we have no idea. Sex would actually, you can also just add extra information about this as well. The group in this case, we say patient, but what kind of disease are we talking about? What was the problem? We don't know. And handedness, we have no idea how this was measured. If you just have your participant TSV. So what I would tend to recommend is one of the tools that several people in my lab are working on and developing is this Neuroscience Annotate tool, which will help you sort of annotate your participant TSV to make sure that anyone who comes to your data after that will have more information on what's actually in it. There's also something similar you can do with your events.tsv. I won't get into too much details about it because it's a very rich sort of framework to make sure you add plenty of very good annotation to your data. And it's called hierarchical event descriptors. And they have data annotation tools that I highly recommend you use to give more details about what participants were doing in their during the experiment. Then if you need to access BIDS dataset out there, well, first of all, you have the BIDS examples, which I think I mentioned earlier, which is a repository with plenty of datasets in it. So there's just a very thin sort of documentation around it where you have a listing of all the different datasets or by modality. And then if you click on any of those datasets, it will bring you to the GitHub repository where you can find it. And that is usually meant for you to sort of browse and get an idea of how to organize similar data. The good thing about this repository is that all the data was actually removed from each file. So each file is actually 0 kilobytes. So if you clone the repository, it's at most maybe like 50 megabytes on your hard drive. So you can always have this thing on your hard drive, and you can always have a look if you want to know how to organize your data. If you just don't want just empty shells when it comes to BIDS dataset, then and you actually want to work with some existing BIDS dataset, the main place to go at the moment that where you can get open data is OpenNeuro. And if you want to sort of search for very specific data, there are 2 ways to do it. If you're interested in just a whole data set, the way to do this, for example, would be to use just the search bar from the OpenNeuro website. But if you are interested in actually more precise sort of search when it comes to OpenNeuro, and for example, you want to know, I want the data set that have anatomical MRI and bold MRI, where each participant had 2 sessions, and I only want you to find the male participants who are between 25 and 35. Then I would suggest you use this other tool from the lab called Neuroscience Query, where you can have where you will not only get the name of the data sets where you can find those participants, but it will also give you how many participants and the identity of each participant in each data set. So that was for BIDS dataset and where to find them. Okay, then now you have your data. Either you got it from OpenNeuro or it's your own and you have to work with it. In general, I will recommend for you to have a look at those three sort of either library or packages, depending on your favorite language of programming language, sorry. So those are like the Swiss knife. Those two are like the Swiss knife when it comes to BIDS. They are meant to have plenty of functions in them to just help you in general to work with BIDS, but their main function is usually to help you find specific data within a data set. Meaning that, let's say that you want to run an analysis, but you only want to start the analysis on subject number 2, session number 2, for the task where the subject was on a localizer, and only the first run. So you can sort of use those tools to only retrieve the specific file and the specific metadata for something like this. And the way they usually work is that they will index all the information and all the data within a BIDS data set, and then you can sort of query them to sort of with specific questions. So you can they will return to you the file names and the metadata that you're interested in. So yes.
I interrupt you for a sec. It's just near the end of the hour. We can go over the hour as far as room booking goes, but I recognize some people might have to leave and just
I'm pretty much done. I have just those few slides to do. Yeah.
Okay.
So I just like in 2 minutes I'll be done.
Okay.
Sorry.
No, we
So yeah, the other one, like when I was on the one hand, I was showing you the very low level tools like to work with BIDS, and then there's also very high level tools that are usually fully automated that are called BIDS apps. And then the way they work is that you give them a BIDS data set and they will run an entire pipeline on it. The most famous ones are, for example, MRI QC to do quality control on your data, or fMRI prep to pre-process your data, but there's plenty of others that are listed on this website here. Okay, then I need to tell you a little bit quickly about BIDS extensions. I think I mentioned them quickly before, but the idea is that your data set, the type of data you work with might not be in BIDS at the moment. But there are BIDS extensions to potentially support it. The way you may know about them is just by checking the link here that gives you a list of all the data type and all the BIDS extension that are sort of work in progress. And the way they usually work is they start just as a simple Google Doc where people are going to discuss how they want to organize this new type of data. And when this thing gets a bit refined, then we might start integrating them into the BIDS specification. So you might just get a website to sort of help you figure out how to sort of organize this type of data. And what I tell people is, if you have a type of data that is concerned or that matches what's in one of those extensions, go and read the extension, because there's already plenty of information and good suggestion on how to organize your data, which is usually going to be much better than you trying to sort of improvise. So those are still very valuable source of information, even if they're not like official BIDS yet. Okay, and so getting very close to the end. So these are like the final pointers in case you need help when it comes to BIDS. What we tell everyone is go on the discourse forum called Neurostars. It's not specific to BIDS, but it's about neuroscience at large, globally, I would say. So, and there's plenty of people there who know about BIDS and many other things, so they'll be able to help you. If you want to get some update about BIDS, there are different ways to do it. The Google group, we just called the BIDS mailing list, is one way to do it, where you get, at most 2-3 emails a month. It's not that active, so you won't be spammed. We try to also post information, post updates on Twitter and Mastodon, so you can also follow us there. And if you have problems, the other places where to go are go directly to the GitHub issues and open GitHub issues on the repository of interest, either the specification, the validator, or some other tools. And then you can also go on Mattermost, which is sort of the open source version of Slack. We use the BrainHack Mattermost for this, and it's in sort of a place with, I think, more than 5,000 people at the moment. Don't worry, it's not that active, so you won't get spammed either, but it's definitely also a place where you can get updates about BIDS. And then if you after answering question, you might also end up helping others, and then you can sort of join the community of BIDS contributors. That's also, I guess, one of the strengths of BIDS is that so many people that use it that it's easier to sort of get help, and also then you end up also then helping others. And how you can contribute, there are many ways. The most obvious one, I would say, is if you have data that is concerned by a BIDS extension, or BEP as we call them, then go and read the extension, make comments on it. Eventually, if the BEP has recommendation on how to organize your data, try to organize your data following what they suggest. So you'll be able to give feedback to the people who work on this BEP. You can work on tutorials. Let's say you struggle with something with BIDS, we can add a tutorial to the BIDS starter kit. If you find bugs somewhere, just make bug reports. That is always super useful. These are some of the ways you can sort of help you sort of work with and contribute to BIDS. And then if you keep on contributing, then eventually people will just ask you to come and join the BIDS maintainers. That's pretty much what happened to me. I was always on the GitHub repository. Eventually they were like, well, you might as well just join our meetings. And now there's sort of more than there's 10 of us in the BIDS maintainers, and there's also the BIDS steering group. So usually the way you describe it is the BIDS steering group is the people who make sure that the BIDS ship goes always in the right direction, and they have their eyes on the horizon, and they always try to keep the big picture in mind. The BIDS maintainers are the people who are just on deck and trying to make sure that everyday things are getting done. And that would be all for BIDS, at least from my side. And I would like to sort of thank you for your attention and keeping up with all of this. And I would like to thank all the members from the Origami Lab, and also the people from the NeuroBagel team, because they are very helpful on making some progress on many of those things. Okay, I'm gonna be quiet now, and if you have a question, I'll be